for slides. And if you want to turn your mics on, I'll start here and then I'll come join you. So for our next session, Christian and I are very happy to co-host and co-moderate this discussion. So as Jan mentioned in his introduction, one of the projects that I think we can honestly say that we are most proud of is the semi-recent meta-analysis project on the phase 3 quinolone trials. And so this is, as I mentioned earlier, a collaboration between the critical path to TB drug regimens as well as the WHO, bringing in the University of California, San Francisco. You'll hear from Radha and her team a little bit later about the statistical analysis plan that she's launched. The intention of this project is really to make use of a very important data platform that we call TB-PACS, which is the data initiative led by Critical Path Institute and the TDR to aggregate these 3 clinical trials and make them publicly available to each and every one of you actually for access. And should mention that partnership is also in alliance with all the 3 different owners of these clinical trials, as well as those 2 institutions that I mentioned. So our focus today is going to be on the presentation as you see here in the outline. Christian and I are just going to do a little bit to set the stage for how this partnership came about. We appreciate that many of you may have heard an acronym here or there, or may have heard here or there that we're working on a meta-analysis program. I think maybe 3 of you were able to join us at the union meeting where we had the initial presentation of the data. Unfortunately, it was not scheduled as well as we would like, and there were a lot of conflicting sessions. So this will really be our very first opportunity to share with all of you as critical stakeholders some of the preliminary findings from that meta-analysis. We will take a very well-scheduled break after Radha's presentation today so that we can prepare for the panel session at that time. So hopefully you can hang in there with us for an hour, hour and a half. So we are going to try and move through the rest of the slides from the couch, so hopefully that will work. This is always the toughest part of the job. This is advancing. Oh, so we had it flipped. There we go. Great, thank you. And will we be able to advance from there? Okay. Okay, so we have acronym soup here in CPTR as we know, and so we, when we talk about TB Reflect, this is really the reanalysis of the fluoroquinolone executed clinical trials, and it added up to a nice acronym, which is TB Reflect for us. This has sponsorship both from CPTR and the World Health Organization, as I mentioned. Having said that, there is a large steering committee that has contributed to both the design of the objectives for this meta-analysis, the design of the statistical analysis plan itself, and then the work is actually being executed, as I mentioned, by University of California, San Francisco with Radha Savik and her team, and she'll be talking to you today in detail. So Christian, if you want to talk us through the goals of TB Reflect.
Does it work? Oh yeah, very good. Thank you very much, Debra. So TB Reflect is born mainly from a discussion we have had, Debra and I. It was, I think, in the Union Conference in 2015 in Barcelona. There has been the publication of the 3 trials on treatment shortening for drug-susceptible TB, including quinolones, either moxifloxacin or catifloxacin. They were published in the same issue of the New England Journal of Medicine. So these are oflotab, Remox, and rifaquin. And the 3 trials didn't show any type of superiority or at least equivalence of treatment of 4 months duration, including a fluoroquinolone, compared to the standard regimen used to treat drug-susceptible TB. So Debra and I were thinking about, these are 3 negative results, as we say. What can we draw from that? And we thought that with the investment that were put into these trials, there were more than 6,000 patients being involved, recruited, and contributing to the trials. A lot of time being invested, a lot of money. We are speaking about, or if we put all together, at least $50 million. So we cannot be satisfied only with the fact that too bad it didn't work. So we thought really we need to do something out of that and try to at least refine the understanding and try to see what does it tell us? What these 3 negative trial results tell us? What can we learn from it? What can it tell us that we are not seeing because we are speaking about 3 individual trials in terms of how to design new trials, in terms of what were the endpoints being selected, and what does it say about moving from phase 2 to phase 3 trials with the proxy markers that are being used, etc. So we are really interrogating ourselves on how we can really make use the best ways possible of this trial. Here came the idea of creating this meta-analysis of the quinolone containing shorter regimens, and with a view, as you can see, of several aspects, to focus on improved understanding of the factors responsible for the variability in the patient's response to treatment, try to understand at the individual patient level what was happening, evaluate the endpoints of treatment outcome for the selection of new regimens to bring broad from phase 2 to phase 3 trials, and optimizing the statistical methods to compare results between the regimens. So that were the goal of the meta-analysis, and we thought that it would be a very good way to start using TB Pacts, which has been set up by CPTR just a little bit before, which is an area, a collective database where trials are being put together for access for further analysis. So the idea was that researchers would develop a framework linking clinically relevant endpoints with the response of the bacteria to treatment. So next slide. Thank you. So what we are thinking of in terms of benefits is that if we do a meta-analysis, putting together the data of the 3 trials and relooking at the way they were analyzed and at the questions that we could ask, then we could have an improved understanding of the source of variability of patients' response to treatment. The other benefit would to have a determined predictable linkage between the pathogen load dynamics and the clinically relevant endpoints in TB trials, try to see how we can relate those. Could these 3 trials tell us more about how the pathogen load dynamics can lead to clinically relevant endpoints in assessment of efficacy? Improved selection of new regimens to be tested in phase 3 clinical trials is also another potential benefit. And then going beyond that, we thought we could have optimized statistical methods for the assessment of non-inferiority, together with incorporation of PK/PD parameters into primary analysis. That's based on the fact that for the first part, the 3 trials were all conducted as non-inferiority trial, and the second part is that some of the trials had PK/PD data that could be used in order to inform better about the distribution of the drugs and how the various dosage and the way that the delivery of the drugs could relate to the outcome that were being evaluated. So these were the expected benefits from the meta-analysis. In terms of potential impact, what was envisioned is that we could look at the data to inform us on what could be best individual dosing of drugs. Also looking at this meta-analysis could inform on the design of studies or design of new trials. Especially there are some movements now in the TB world about establishing new trial design. We are speaking about having a phase 2C type of trial between the phase 2B, the classical phase 2B, and the phase 3 trial. So how does it help selecting new regimens early that can be brought from phase 2 to phase 3 trial? And then, of course, looking at mechanistic models of pathophysiological process. So we thought that with that, the TB drug development field, relying on experience and on expertise, could really enter the 21st century by applying modern approaches, technology, and resources. In fact, there was a seminar at the WHO a couple of weeks ago with Trish Glover, who is the editor of the BMG Open. And she gave a talk, an interesting talk on negative trial results. And she really said how much the absence of publication, which was not the case, fortunately, for the equivalent trials, but the absence of publication of the negative trial result is a waste of resource and a waste of information. And the fact that the scientific community doesn't take more advantage of the negative trials to really inform the future development of drugs and regimen is really of a concern. So I thought that was resonating very well with what CPTR and WHO has decided to do. And we place ourselves reasonably into this frame of learning from experience to inform for the future. So the TB Reflect collaborators are here listed. These are the people who have expressed interest, and major part are the PIs and investigators of the 3 trials that were invited to work together on what will be the design of the meta-analysis. So we welcome really all their contribution. We have extended to other scientists who are interested in helping focusing on the analysis, but altogether this forms the steering committee that has been meeting twice in order to really influence and orientate the analysis, but this is an ongoing process, and there will be further opportunity to be working together.